Alright, today what we're going to talk about is eukaryotic transcription. The themes are very similar to the prokaryotic transcription, but you're going to see that there's going to be many more regulatory steps involved and many more players that will be involved. Okay, so in the last lecture we stopped with the information that in eukaryotes there are three types of RNA polymerases. They're named RNA polymerase 1, 2, and 3. So how were these polymerases actually found? It was found through biochemistry. So what the researchers had done was they took large cell extracts. An extract means you take the cells and you burst it up and release all the ingredients, all the proteins, etc. And then what they did is they separated the proteins in these extracts based on some type of properties such as size, shape, etc. through certain DNA, certain protein separation columns, right? So like gel filtration column would separate proteins based on their sizes. So all the protein complexes in the extracts were separated based on their sizes, and so what happened basically is this. So what you do is you take so let's say this is your column, it's full of some material that's going to prevent the protein's migration through this. You're going to put your extract up in here, and how fast the protein complexes will go through this matrix, through that column, is going to determine when the proteins are going to come out, right? So after you load your proteins and wait for them to go through the column, what you can do is you can collect fractions. So these are called fractions. So what you can do is you can collect a little bit. Let's say you collect 100 microliter of the volume containing the proteins. Let's say this gel extraction column is separating the proteins by size. The first fraction, what type of proteins will that contain? Smaller protein complexes or larger protein complexes? Smaller, right? Because the smaller things are going to go through the gel. It's just like DNA gel electrophoresis, right? You are separating molecules based on some physical property of theirs, right? So the small guys are going to come out first. The first fraction, you're going to put it away, then you're going to collect your second fraction and third fraction, etc., etc. So your different types of protein complexes are going to be in different fractions. Does that make sense? I think you went through some biochemistry at the beginning of the lecture. All right, so that's what's plotted here. So what the researchers did is they took each fraction and they looked for if that fraction can support RNA synthesis, right? So they mixed the fraction, the proteins in that fraction, mixed it with some DNA and some ribonucleotides, and looked, can it support protein synthesis or not, right? And that's plotted in the y-axis in pink. So what they found is that there are 3 peaks, distinct peaks in these fractions that could support RNA synthesis. And the reason for that is each of the RNA polymerase complexes are different sizes, and they were found in different fractions, and that's how they could identify them. So these protein complexes containing RNA polymerases 1, 2, and RNA polymerase 3. Okay? So that's what plotted here. Also, there is further what the researchers did is they knew that there was a mushroom toxin called alpha-amanitin that was capable of reducing transcription in previous experiments. And when they added this alpha-amanitin in different fractions, what they saw was that the transcription by the fraction containing polymerase 1 was not affected, so that's plotted in blue here, but the transcription containing fraction containing the polymerase 2 was affected. That suggests that the alpha-amanitin specifically inhibited the transcription activity of polymerase 2. Okay? So that was the initial biochemical characterization of all of these polymerases. And later, we know that these polymerases, later experiments show that these polymerases are similar in structure, but they do interact with different sets of proteins, and they're specialized to transcribe different types of genes. So the RNA polymerase 1 transcribes RNA genes. Pol2 is mostly for mRNA, so protein-coding genes. And Pol3, the major class of genes that Pol3 transcribes is tRNAs. All right. So here is the structure. The overall structure of RNA polymerases are similar. So here's the structure of the bacterial RNA polymerase shown here. You learn this beta, beta prime, alpha, and the omega subunits. And the eukaryotic polymerases, in this case, we have a structure for the eukaryote, the single-cell eukaryote yeast. The polymerase looks in shape very similar. You have two large subunits making up the catalytic core, but there are many more protein subunits interact with this core complex. In addition, there is a specific domain in some eukaryotic polymerases, RNA Pol2, in the large subunit, in which a C-terminal domain, there is a tail that sticks out here. Okay? So that's the C-terminal domain. And the C-terminal domain contains of 26 to 52 repeats of the same amino acids. So you have the same 7 amino acids repeated many, many, many times, and that is the C-terminal domain that sticks out of the large subunit of RNA polymerase. So here's the sequence of that repeat. So C-terminal domain repeat. These are the 7 amino acids, and you can see that it contains 3 serines, a threonine, a tyrosine, etc. And what later the researchers found was that some of these amino acids could be phosphorylated. So there is a specific phosphorylation of serine 10, serine 5, and serine 7. So this is actually a distinguishing feature of eukaryotic RNA polymerases from prokaryotic ones. So what does the C-terminal domain do? Okay. So first, the researchers figured out, okay, these C-terminal domain are phosphorylated. So they said, does that have anything to do with transcription? So to look at that, they wanted to look at interface chromosomes, because during mitosis, chromosomes are highly compacted and there is no transcription. So you have to analyze transcription in interface chromosomes. But as you can remember, the interface chromosomes were very decondensed, and you couldn't see very well under the microscope for a certain specific chromosomal site if there is transcription or not, right? So you need some interface chromosomes that are visible, that you can see a certain site and distinguish between two conditions where that site is being transcribed versus not transcribed, if you want to look at anything that has to do with transcription. And for this, they have used a special type of chromosomes that are isolated from Drosophila salivary glands. These are called polytene chromosomes. These are special because they are interface chromosomes, but you can see them. And the reason why you can see them is that each of these chromosomes get replicated many, many, many times. But after the replication, the cell doesn't divide, and each of these chromosomes keep aligned next to each other. So each bundle that you see here contains many, many copies of the same DNA, the same chromosome. Does that make sense? It's like a stack of spaghetti noodles, okay, right next to each other. Does that make sense? Okay. So what that makes them large enough to be able to look under the microscope. All right. So these are the polytene chromosomes. The second thing that they knew was that certain sites on the polytene chromosomes sometimes puffed. Okay, so that puffing is due to the decondensation of the chromosomes because those sites are transcriptionally active. So you can see here, this is the same chromosome in a condition where the genes in regions A1 to, so this is these are the bandings, and these bands are stereotypical, so you can see these on polyethylene chromosomes, and the bandings help, the bands help you determine what chromosomal region you're looking at, okay? So what you can see is that here's a chromosomal region in chromosomes that where the transcription is inactive, and here is the same chromosomal region where a bunch of genes in this chromosomal region is transcriptionally activated. What you can see is a drastic difference in the structure, in that when there's active transcription, the polyethylene chromosomes make a puff. And that puff is due to the decondensation, because transcription needs the chromatin to open up so it can be transcribed. Okay? So, what did they do? What they did is first they made some specific antibodies. So the antibodies are amazing molecules, and they are used in molecular biology because they can distinguish very small differences in their antigen, right? So one can actually take the C-terminal peptide, chemically synthesize it, and then inject into goats. And for goats, this peptide is going to be foreign, so it's going to make antibodies. And it's going to make antibodies that are specific to this unphosphorylated CTD peptide that you injected. Okay? So you can take the CTD peptide, again, now phosphorylate some of the residues, just like they are phosphorylated in vivo that you know, right? So like serine 2, serine 5, etc. And then inject these into a rabbit. Then the rabbit is going to make antibodies that are specific to this phosphorylated CTD peptides, okay? So these rabbit antibodies are only going to bind to the phosphorylated CTD, and the goat antibodies are only going to bind to unphosphorylated CTD. Does that make sense? Good? All right. So then what you're going to do is you're going to isolate your polyethylene chromosomes. You're going to put it on a slide, and you're going to throw in those two antibodies. And what these antibodies are going to do is they're going to bind to their targets. So then you're going to use secondary antibodies. In this case, the secondary antibody for that will recognize the heavy chain of the rabbit antibody is going to be conjugated to some red fluorophore, and the secondary antibody that is going to recognize the goat heavy chain is going to be conjugated to a green fluorophore. Okay? So wherever you see a green signal here is going to tell you that there is some RNA polymerase there with a C-terminal domain that's not phosphorylated. And where you see a red signal is going to tell you that there is RNA polymerase there with a CTD that's phosphorylated. And what can you tell in terms of these puffs here? What can you tell me in terms of these puffs? Where you see the red signal. Is there high transcription or low transcription?
High transcription.
High transcription, right? So where you see high transcription, what type of RNA polymerase do you see? C-terminal domain phosphorylated or not phosphorylated? Phosphorylated. So then from this we learn that the C-terminal domain phosphorylation is correlating with transcriptional activity. All right? All right, so these two regions actually were molting hormone. So these two regions were encoding for genes that respond to the molting hormone. So they puff because they get transcribed. All right. So the type of phosphorylation that occur on the C-terminal domain of eukaryotic RNA polymerase is different at different stages of transcription. Just like bacterial transcription, in eukaryotes we have transcription initiation, elongation, and termination. So if you look at the types of phosphorylation that occur at these three different stages, you actually find that when the RNA polymerase 2 is sitting at the promoter in a pre-initiating complex that is a closed complex, the RNA pol 2 CTD is non-phosphorylated. Once the RNA pol 2 starts initiating transcription, the the serine 5 at the CTD becomes phosphorylated. When the RNA polymerase transition into an elongating state, an elongating transcription, an additional phosphorylation site, which is the serine 2, gets phosphorylated, and towards the end, the serine 5 gets dephosphorylated, and you have RNA polymerases with CTDs that are phosphorylated at serine 2. The way I remember this is that 5' of the genes have serine 5 phosphorylation, and the serine 2 is the downstream. Okay, so if you ever want to remember. Okay, and the role of these different phosphorylations is basically acting like specific binding sites for other proteins to bind and do their work. For example, serine 5 phosphorylation at the beginning of transcription serves as a binding site for some proteins that are important for the capping of the RNA at the 5'. Okay. Any questions about this? All right. So that was one of the distinguishing features of prokaryotic and eukaryotic RNA polymerases. Now, the second distinguishing feature is the complexity. There's a lot more complexity in eukaryotic transcription, and that's reflected not only the numbers and the types of proteins that are involved in transcription, but also the type of DNA sequence elements that help regulate transcription, right? So if you look at a typical prokaryotic promoter where you have an ORF, that stands for open reading frame, right? That is the sequence that is that codes for the protein. Upstream of that ORF, you're going to find your -35 and -10 sequences, with or without an operator where the repressor is going to bind, and maybe some other sequences where your transcription activators are going to bind. In a simple eukaryotic organism, the gene regulatory elements are again found upstream of the transcription start site. Here is +1. And these elements can be found at further distances. And in addition, you're going to find additional elements which we call enhancers, which are a little bit less frequently seen in prokaryotic transcription. Okay? So you go from yeast promoters to human promoters, and you're immediately going to recognize the number of the elements that are important for transcription have increased, and their distances from the promoters also increased. Okay, so what you have here is a mammalian gene. A +1 is the transcription start site, and right upstream of the +1, you have a sequence that goes includes the TATA, which is appropriately named the TATA box. And you have other sequences that are important for RNA pol2 binding to the promoter. These are called promoter proximal elements because they're close to the promoter, so they're proximal to the promoter. You have these exons, you have introns, and you have enhancers that can be anywhere pretty much in the vicinity of the gene. It could be it could be in the intron, it could be downstream of the gene, it could be upstream of the gene. It could even skip some genes and be away from the actual gene that it controls. So what we call this region here in mammalian genes are the basal promoter. It can include a TATA box, sometimes it doesn't. That's where the RNA polymerase complex assembles. Okay? So you have these promoter proximal elements that are involved in tethering the complex to the promoter, RNA polymerase complex to the promoter, and then enhancers are more distal elements. Distal means far away. What they do is they control tissue-specific and temporal expression of a gene. All right. In mammalians, there are two types of promoters. One is a TATA-containing promoter, so it includes that TATA box. And a second type of promoter is called a CPG island promoter gene. Okay? So these genes do not typically contain a TATA box. Instead, what they contain is a stretch of DNA region that contains lots of CG repeats. Okay? CPG simply means there is a C and a G next to it. Okay? So these CpG ion promoter genes make up about 60% of the promoters. They're usually transcribed at a lower level than TATA-containing genes. So the TATA-containing genes are transcribed much higher, and CpG-containing genes transcribed at a lower rate. All right. In vertebrates, we learned in the DNA repair lecture that in the CpG containing sequences where you have a C next to a G, right? And a lot of these C's can be methylated, all right? So the DNA gets modified in that there is a methyl group that's added to the cytosine. So that's cytosine methylation. So in vertebrates, there are a lot of C's that are methylated. But because that these C's can mutate, right? So there are the mutations converting methylated C's. There is less CpG sequences than expected in vertebrates. A lot of these C's are converted to T's. Anyone remember why cytosine methylation can be mutagenic? You guys studied the lectures, right?
Deamination.
What is it? Deamination. Because when it deaminates, it's going to convert into a T, and if the repair pathway doesn't catch it on time, it's going to be converted in the second round of replication. It's going to mutate. Now, throughout the evolutionary time scale, which is going to be millions of years, right? For each species, vertebrate species, the number of C in the genome is going to reduce. Now, that happens in a lot across the whole genome. So when you're looking at the whole vertebrate genome sequences, you're going to see that there's a CpG deficiency, and within this genome, you can actually find what we call CpG islands. Okay, so these are somewhat arbitrarily defined as being about 1,000 base pair in length with elevated CG composition. So there is little CpG depletion, and frequently you will see less cytosine methylation. At these regions, you're going to see less methylation of the cytosine. So they act as promoters, but they have less precise transcription start sites. For a lot of the TATA-containing promoters, the transcription start site is exact. Usually there's one start site, that's where the transcription starts. For CpG-containing promoters, the transcription start site is less exact. That means there will be multiple transcription start sites at the promoter. There is also divergent transcription. This is interesting because what will happen is that the same CpG-containing promoter is going to position RNA polymerase to start transcription in one direction. It can also do so in a way that the transcription will be started on the other direction. So in a lot of CpG-containing human promoters, what we see is that transcription is started in both directions. Now, the transcription that starts in the correct direction is going to make that gene, right? The transcription that starts in the incorrect direction, the function of that transcription is not very clear. What we know is that a lot of the RNA that is made, the RNAs are short, so while the RNA polymerase starts transcription in that direction, it doesn't necessarily go further. And a lot of those RNAs gets degraded, so it doesn't get used. But why are they transcribed? We still don't know. That's still an open question. All right. Any questions so far? Good. So what are the actual sequences of the basal promoter elements? We talked about consensus sequences. So these are the consensus sequences. This is the consensus sequence of a TATA box. So it contains T, A, T, A. And in the next location, you can have A or T following with an A. In the next location, you can find an A or a T or an A or a G, right? So this is another way to write a consensus sequence. There is another sequence element that's important for transcription in Drosophila and mammals, and this is called the initiator sequence where you have this sequence here, and the downstream core promoter element is a consensus sequence here, and a TF2B recognition element here. These Y's and N's, N stands for it can have any sequence at all. It just needs to have a nucleotide here, so that spacing is important. And when you see a Y or another notation, these say any purine or any pyrimidine. Okay, so it could be A or a T or a G or a C. All right. So how are these sequences recognized? In eukaryotes, the assembly of the pre-initiation complex. Pre-initiation complex is the RNA polymerase with many of its initiation factors coming in and binding to the promoter and ready to go. That assembly of the pre-initiation complex is a multi-step process. Okay? It involves a lot of steps, and it involves recognition of a lot of those basal promoter sequences. It involves the sequential recruitment of the initiation factors. How does it all start? We know this much better for TARA-containing promoters. So what we have is that there is a protein called TBP. It is named for TARA binding protein. This protein has that kind of nice little shape. What it does, it binds to the TARA box, and it does so in the context of a protein complex called TF2D. So the rest of the TF2D subunits are not shown here, but TF2D also contains TBP. So what it does, it binds to the minor groove of the DNA at the TARA box, and that binding bands the DNA. So it bands the DNA like this. Okay. Next, there are two other proteins called TF2A and TF2B that bind to DNA on either side of the TBP. So the TBP binding recruits these proteins. How a protein recruits another protein? It recruits so if it has protein surfaces that can interact with the other proteins, right? So that's what we call, when I say recruitment, that's what I mean. All right. So you have this TBP, TF2A, TF2B, and those proteins, what they do is they recruit the RNA pol 2 TF2F complex. TF2F is a heterodimer of two proteins, and with RNA pol 2, it binds, recognizes this TBP complex at the promoter, and it binds. So RNA polymerase 2 binding to the promoter happens, right? And it's stabilized by the TBP, TF2D, and the other initiation factors. And those factors position the RNA polymerase over the start site. After the RNA polymerase is positioned over the start site, there is another protein complex called TF2E that interacts with the RNA polymerase, and it creates a docking site for TF2H. And this is a 10 subunit protein complex that binds to the whole RNA polymerase. All right, so we have 2F2H coming, and it binds to the RNA polymerase. And this whole set of proteins with the RNA polymerase sitting on the promoter is what we call a pre-initiation complex. Okay? This pre-initiation complex has TF2H, and TF2H has a helicase activity. It uses ATP to open up the DNA, double helix, to form an open complex, right? It's just like the bacterial transcription. You went from being a closed complex to an open complex, and in this case you used TF2H, the helicase activity of TF2H, to open this up. What does this remind you in prokaryotic transcription? There was a special case in prokaryotic transcription where the sigma factor, which is the initiation factor with RNA polymerase, sitting at the promoter but not initiating transcription because it needed interaction with another protein which helped with ATP to open the DNA. Do you guys remember? Bacterial enhancer? Remember the transcription factor NTRB or C, I can't remember now, that interacted with the polymerase and sigma 54, and the energy from that interaction opened the DNA strand so that transcription can start, right? It kind of reminds you the types of steps in prokaryotic transcription, but it's a lot more elaborate. Does anybody remember this enzyme from anywhere else? TF2H. Did you guys review the DNA repair lecture? Not good. I'm not going to give you the answer, so you have to go and check. All right. I'm going to make you study. Okay, so what happens next? After the pre-initiation complex assembles, you have an open RNA polymerase complex where the two DNA strands are opened. You have a TF2H complex also phosphorylating the serine 5 at the C-terminal domain, which serves as a docking site for the 5' capping enzymes. So after the protein complex assembles and starts elongating, a lot of these proteins that I talked about, right, that are assembled in the pre-initiation complex gets released, okay? So these are called initiation proteins because they get released afterwards. So those most pre-initiation proteins disassemble as pol 2 starts elongating, and that pol 2, when it starts elongating, it interacts with other set of proteins called elongation proteins that help the RNA polymerase to elongate along the gene. Which factor in prokaryotic transcription disassembles after the polymerase starts elongating? Come on, this is an easy one. There is a protein that is a transcription initiation factor in bacteria that gets disassembled after the polymerase starts elongating.
The sigma factor?
Excellent. Sigma factor. That's the one that gets released, right? So it's a prokaryotic transcription initiation factor. All right. And before I start that, any questions about this? Try to remember what are the types of steps that need to happen. Like TBP is an important step. It needs to bind to TARA and bend it, right? TF2H, H stands for helicase. It has a helicase activity. But you don't need to memorize TF2A comes in before B, blah, blah, blah. It's not important, but really important things that you need to remember is what has to happen for the RNA polymerase to come to the promoter. Okay? There are many steps involved. Some of them are key steps, like the TBP binding to the TARA box. All right. So now what I'm going to show you is I'm going to show you a little movie, if I can start it. Come on. All right. So this is a movie that is made from the crystal structures of all of the RNA polymerase subunits that are so far crystallized. So this is the RNA polymerase itself without the DNA. Okay, so you have the RNA polymerase here. And what you have here is the TBP protein bound to the DNA that is the blue here, right? And that bands it. And you have a TF2B protein here in light blue that you can see the TF2B. And it has some structures that actually extends all the way inside the RNA polymerase, the large subunits, towards the catalytic core. So this is the catalytic core where you have a magnesium ion. Okay? So what happens is the TBP, after TF2B, after it inserts close to the catalytic core, you have the opening of the complex the DNA duplex. So you have this transcription bubble, okay? And the magnesium ions at the catalytic core help the catalysis of the first RNA synthesis. So RNA is shown here in red. So once the RNA starts to get synthesized, the first few steps in the initiation involves DNA scrunching. So it scrunches the DNA, opens up the DNA ahead of the polymerase, and making the bubble a little bit larger. Okay? So now you have synthesis of that RNA coming in here, and this is the initial transcribing complex. Now let's remove the initiation complexes and go into the elongation form. Okay? So in the elongating form of the polymerase, you have the RNA that is being transcribed, and it starts exiting the polymerase through a certain channel. Okay, so it's still making lots of contacts here, as you can see, but it's exiting. So these other proteins called elongation proteins can bind to elongating RNA polymerase only because at the initiating factor, these regions of the RNA polymerase was bound by something else. So now we're zooming into the catalytic site where you can see this bridge helix, which is important for the catalysis. And you're going to see these two helices here that are important for catalysis as well. These are called the trigger loops. And this is the magnesium ion, and this is the RNA, and this is the DNA template. All right. So at the beginning of the catalysis, you have these helices, the trigger loops, standing away a little bit, and you have the nucleotide coming in. And when the nucleotide binds, this triggers a certain structural change where the loops now push the nucleotide closer, and the magnesium helps make the bond that adds the RNTP to the growing chain of RNA. After the addition, the trigger loop backs a little bit, and the whole DNA translocates, right? So it gets to the next, it gets pushed so that the next nucleotide can act as the template. Again, you have the new RNTP coming in, and the loop is going to push and close the active center. So this is going to get inserted. You're going to have your bond made, and the pyrophosphate here is going to be released, and that release actually causes the shift in the trigger loop. So the loop is going to back off a little bit, and it's going to start again. So sometimes we refer these protein complexes molecular machines, and I think this is a beautiful illustration for what we call the molecular machines, because you can see that everything is physical, right? In biology, everything is chemistry, everything is physics. You can see the molecules actually moving and doing their job. So what happens when there's a mismatch? The mismatch gets recognized, and a couple of nucleotides get cleaved. So the complex backtracks so that the mismatched nucleotide is here. A couple of nucleotides get cleaved and removed. And it doesn't stop there, so RNA polymerase get actually arrested. So to recover from that arrest, the polymerase complex does something interesting. What it does is it backtracks. So it goes back and now gets that newly synthesized RNA, translocates it into a new site on the RNA polymerase complex, so where the RNA is now interacting with these proteins. Okay, so this is the RNA backtrack site, and that kind of arrested complex is a very good binding in a very good binding conformation that brings another protein called TF2S. TF2S is an endonuclease. So what it does is it cuts the backtracked RNA. Just gonna endonucleatic cleavage. That RNA is gonna be released and degraded, and the RNA polymerase is going to be rescued. Okay, so that pausing is gonna stop, and it's gonna rescue itself and continue the transcription. Any questions about the movie?
What do you mean by RNA arrest?
RNA polymerase arrest? RNA polymerase, once it starts transcribing, it's going to move, right? Or it's going to pull. So it's a matter of debate. It's relative. But it's going to move one nucleotide at a time. It's going to add, translocate, add, translocate. Arrest means stop. It's going to get arrested. When there is a mismatch, it's going to stop because it made a mistake, right? It's kind of like the DNA proofreading mechanism. Any other question? All right. So for many, many years, researcher and for the past 40 minutes, us completely ignored something, right? What did we ignore? We ignored the fact that eukaryotic DNA is not naked in the nucleus. How do you find eukaryotic DNA in the nucleus? What's the basic unit of chromatin? Nucleosomes, right? The DNA is wrapped around nucleosome. And that wrapping around the nucleosome is actually inhibitory to transcription. The transcription literally requires the DNA to be naked, opened up, and then you use one of the strands as a template to make the RNA, right? You're not going to be able to do that in the presence of a nucleosome. So how do we deal with nucleosomes during transcription? So this is kind of illustrated here. You have nucleosomes around here. You have all your initiation proteins. They need to bind to the DNA, but if they are wrapped around nucleosomes, they're not going to be able to do that. So how does that happen? So the opening up of chromatin is mediated by transcription activators in eukaryotes. Okay? So here's an example transcription activator called GCN4. In eukaryotes, transcription activators, proteins have typically two domains. One is called the DBD, DNA binding domain. The other one domain called the AD because it's called an activation domain. It activates transcription. So the DNA binding domain binds to upstream activating sites in the genes that GCN4 regulates. So the GCN4 activation domain interacts with a group of proteins that make up a complex called SAGA. Okay, so this is one example. It is called a chromatin modifying protein complex because it has a subunit called GCN5 that is an acetylase that acetylates the histone tails around where it binds. Okay? So where the acetylation of the tails occur, if there's hyperacetylation, hyper means lots, right? And hypo means less. So hyperacetylation of the N-terminal tails occurs through bringing this complex containing GCN5 that puts lots of acetylation. Now, acetylation of the tails actually loosens up chromatin a little bit. So it opens the chromatin. It negatively affects the compaction of the chromatin. In addition, the acetylation of tails serve as binding sites for other proteins to come and recognize the promoter. So one of these proteins, in fact, is actually a transcription initiation complex. So this is TF2D. It has subunits in itself that can bind to acetylated tails through these domains called bromodomains. Okay? So it recognizes these acetylated sites and it goes to it. All right. The acetylation also helps another protein complex that is called switch SNF, which also has subunits in itself with bromodomains that bind to acetylated tails. Now, switch SNF is an ATP-dependent chromatin remodeling factor. It literally takes the histones out of the DNA and removes the nucleosome. Okay? So the GCN4 transcription activator recruiting the acetylase complex, acetylating the histone tails at the promoter regions, mediate not only bringing in TF2D, but also removal of the nucleosomes. Does that make sense? This is important. This is very conceptual, right? You don't need to remember the names of the proteins, but you need to understand what are the types of steps that are important for the promoter to be cleared off so that you can actually assemble this pre-initiation complex. what happens when you actually want to repress transcription? So in eukaryotes, repressing transcription can occur through chromatin. So what you do is you need to open chromatin for the transcription to occur. You can close the chromatin so that the pre-initiation proteins cannot come and assemble at the promoter. So how does that occur? It's very analogous to what happens to transcription activator. In this case, instead of an activator, we have a transcription repressor. Okay? So this is another example in yeast, where you have the UME6, which is a transcription repressor. The repressors, just like activators, typically contain two domains. One is a DNA binding domain that specifically recognizes certain sequences and binds to them. And the other one is called a repressor domain that is important for the repression. So the DNA binding domain of the repressor, UME6, binds to an upstream repressor site 1 in some of the genes that it regulates. And that binding stabilizes the repressor at the promoter. And the repressor domain now interacts and recruits a group of proteins that make a repressive complex. It is called, that involves a protein that itself is a histone deacetylase. Okay? So this is called histone deacetylase. The short version of saying that is a HDAC. Now, the histone deacetylase in this repressor complex is going to remove the acetylation from the tails of the nucleosomes at the promoters. And what this is going to do is it's going to prevent TF2D binding. It's going to prevent SAGA complex binding there, so that you basically stabilize the nucleosomes at the promoter. And once you have the nucleosome stabilized at the promoter, it's going to prevent the other proteins binding. So in this case, you have the Tata box wrapped around the nucleosome. Now this Tata box is not available to bind to TBP. TBP cannot bind to this DNA when it's wrapped around the nucleosome, so the transcription is going to be repressed, right? All right. Here's a question. This is a perfect example of what we went through in the chromatin lecture a couple of lectures ago. Okay? So we talked about the readers and the writers and erasers of histone modifications, right? And here in the past two slides, if you have those two slides, take a look at them on your screens or printouts. Look at the protein names and look at what they do and tell me which proteins is the writer, a reader, an eraser of the acetylation in yeast. Some of these proteins are listed here. What protein is the writer of acetylation? It's in the previous slide. What protein writes the acetylation? Perfect. That is the enzyme that puts the acetylation, so that would be the writer, right? Okay. What proteins read acetylation? What are the readers here? Yes. And SAGA. It's called SAGA. Perfect. TF2D and SAGA are reading that acetylation, right? Acetylation is like an information. It's put down by the writer, and it's read by these proteins that are downstream effectors. Downstream effectors means you read something and do something downstream of that process, right? You watch the news and then go, something like that. All right. What is the eraser here? Because the information can be erased. It's reversible. What's the eraser protein? RPD3. Very good. So RPD3 is the eraser because it's the histone deacetylates. It deacetylates. It removes the acetyl groups from the nucleosome. And this is the type of question that I typically ask in midterms too. So study what I'm talking about now. All right. So in eukaryotic transcription, there is an amazing player that is important for transcription initiation, which is called the mediator. Okay? It mediates interactions between transcription activators and the repressors and the RNA polymerase. What it does is it's a large protein complex made up of multiple subunits, and it interacts simultaneously with the transcription activators that are bound at distal enhancers, as well as the proteins in the RNA polymerase and at the promoter region of the gene. So it transmits the information from the enhancer to the polymerase, and that mediates the transcription. How does mediator look like? The mediator looks something like this. A lot of groups have been trying to crystallize it or use a microscopy to model it. And here is the yeast mediator. And here in blue, it has a head and a middle and a tail domain. It looks something like this. You have the RNA polymerase complex shown in gray and gray here. And then you have the DNA that is shown in orange. So it basically literally encapsulates the RNA polymerase, and then it makes interactions with the activator proteins all over these surfaces. So in eukaryotes, the mediator complex is very well conserved. The Cerevisiae mediator has multiple subunits that are shown here, and the human mediator is very similar. It has many of the homologous subunits, as well as some additional ones. And the idea is that these subunits are specific subunits, make specific connections with certain transcription factors and relay the information to RNA polymerase so that it can assemble on the promoter. All right. So let's now review so far what we've learned. Okay? So in eukaryotes, the DNA is in a chromatin form. So it can be pretty closed and compact. The first, what you have is you have the transcription activators binding to their binding sites, and then bringing chromatin remodeling complexes. These chromatin, we call them chromatin co-activators, so they activate together with the transcription activators, which acetylate the nucleosome, the histone tails in the nucleosomes at the vicinity of the promoter. And that acetylation mediates the removal of nucleosomes at the promoter. And after the promoter nucleosomes are removed, you have the transcription initiation factors assembling at the promoter along with the RNA polymerase, and the mediator complex interacting with both polymerase and the transcription factors and stabilizes this RNA polymerase binding at the promoter, therefore mediating the transcription activation. It's important for transcription activation. So next, what we have is that the assembled pre-initiation complex is going to start transcription, and when it does that, a lot of the pre-initiation proteins are going to be released, and the polymerase, the elongating polymerase, is going to be released from the mediator, and it's going to start elongating the gene. And you can see the RNA, the nascent transcript. Nascent transcript means the transcript that just got made. So that's the RNA that just got made sticking out of the RNA polymerase, and RNA polymerase started elongating.
I just have a question about some of the diagrams. Are the activators and repressors always binding to DNA that's not wrapped around the histone?
Most of the cases, yes. But there are activators and repressors, which we call them pioneer transcription factors, that can bind to DNA sequence specifically, even when the DNA is wrapped around the nucleosome. We're gonna talk a little bit about them when we are gonna talk more about how transcription factors themselves are regulated. But most transcription factors bind to DNA when the DNA is not nucleosomal, when it's open. Any other question? All right. So here is another curiosity of transcription happens. And this has been recognized only about 10 years ago. Okay? In eukaryotes, you have the pre-initiation complex. You assemble the RNA polymerase. all the complexes, RNA polymerase starts elongating in towards the gene, but sometimes it stops. Okay? This occurs at a large number of genes in humans. It was first recognized for heat shock genes in Drosophila. Heat shock genes are genes that are important for response to shifting flies into a higher temperature, and then realize that this is something that occurs more than half of the genes in humans as well. Okay, so what happens is RNA polymerase stops. What we call that is pausing. It pauses after it elongates a certain number of nucleotides. It stops, and that is regulated by this protein called the negative elongation factor. So if it interacts with the RNA polymerase, it negatively regulates elongation, so it stops the elongation, so the RNA polymerase stops. Then, if there is a cue from the cell that phosphorylates, there is a protein that phosphorylates the negative elongation factor, and that gets released, and now the RNA polymerase can continue transcription, and it starts elongating, and it doesn't pause anymore. Okay, so this is called RNA polymerase pausing, and the gene gets transcribed. Can you tell me? Imagine, right? I didn't tell you the answer of this. Imagine what could be the function of pausing in eukaryotes. Why would you have this pausing mechanism?
Make sure it's assembled correctly, so when it continues, it works right.
That's one, yeah. But you could have another mechanism that if it doesn't assemble correctly, you never start transcription. So why start and then stop, right? It doesn't really quite make sense. Why would you want to do that? You assemble, do everything, and polymerase comes in, starts and stops. Waits for a signal. That's the key. You want to take a guess?
Maybe it's if you don't need any more of the protein that you will eventually produce, you just want to stop all.
You're on the right track. You want to respond to a signal precisely, right? And you want to, if you want to respond to that signal in a short period of time, maybe that pausing is going to give you that advantage, because you have seen, and maybe you have been overwhelmed by how many numbers of things need to happen during pre-initiation, right? There's going to be so many proteins that needs to come in order for this complex, the pre-initiation complex, to assemble. And that actually takes time, right? So if the signal is feeding into the initiation, it's going to take some time to respond to that signal as the transcription of the gene. But if your signal is feeding to a regulatory step where everything is done, all you need to do is release, then your response is going to be shorter, right? And you can see that all of these steps are regulatory. That means there might be mechanisms that can regulate each step, make it slower or faster, and then have a contribution to how fast or slow the transcription of a gene is going to be started, right? So that's the idea, that people think that that's what's going on. All right. Now the RNA polymerase 2 starts transcribing in elongating along the gene. So what you have is this structure here, which we saw so many times so far. You have the DNA here, you have a transcription bubble, you have the RNA, and there's a clamp domain of the RNA polymerase 2 that clamps over, so it holds it. Okay? So this clamp domain basically rotates and closes over the incoming DNA. While it's elongating, they basically holding it, so it has a high processivity. We talked about this in the previous lecture. It's particularly important for RNA polymerase in larger genomes to have high processivity because, for example, in humans, the longest gene that is known is about 2 megabase long. It's huge. And the RNA polymerase, the speed is constant. Okay? So it's an enzyme. It can only do so many nucleotide incorporation in so many minutes. So the speed of that is basically the same pretty much. So that speed is about 1 to 2 kb a minute. So if you just calculate this, for that longest gene to be transcribed, to make one transcript, you basically need the RNA polymerase transcribing continuously for 1 day. That's an amazing ability for an enzyme to stay in and constantly transcribe the DNA into RNA. Okay, so that high processivity is supported by the structure of RNA polymerase that holds the DNA tightly. All right. So what happens during elongation? Again, we totally ignore nucleosomes, but we can't, right? The nucleosomes are going to be there. So you have your elongating polymerase, and in this case, the polymerase is bound by proteins that we call elongation factors. What should be done in front of the nucleosomes? What should be done to the nucleosomes ahead of RNA polymerase II? They should be removed. Otherwise, you can't go through, right? So, and how does that occur? It occurs by these elongation factors recruiting protein complexes that is going to just disrupt these nucleosomes, and also the activity of RNA polymerase. It pushes onto the nucleosomes and removes them. It's a combination of both. And what is happening, at least some experiment in yeast suggests that as the elongation occurs, the nucleosome in front of the RNA polymerase is removed and redeposited in the back of the RNA polymerase. Okay. So now the RNA polymerase is happy, moving on, comes to the end of the gene, and there is some transcription termination sequences that make this RNA polymerase pause. So the pausing at the poly A site on the template, in combination with a low amount of serine 5 phosphorylation and high amount of serine 2 phosphorylation at the C-terminal domain, helps recruit a whole bunch of proteins here. Don't worry what their names are. It recruits a whole bunch of proteins that basically cleaves the RNA and then puts the poly A tail, and that then the transcription is terminated and this RNA polymerase gets removed from the template. I didn't put too many details. I don't want to go into too much detail in termination because, in fact, we don't know that much about termination. A lot of gene expression studies have been really focused on the initiation because we know that's a step that's heavily regulated. Whether or not you want to transcribe a gene is really important for the identity of the cell or the gene regulation. But we find that transcription termination can also be regulated. So in the future, I'm sure the people will learn more about the transcription termination. All right. So this was RNA polymerase II. Any questions about RNA polymerase II transcription? Nothing? Okay. In eukaryotes, the two other polymerases, RNA polymerase I and RNA polymerase III, transcribe different genes. In the case of RNA polymerase I, it transcribes the 18S and the 28S RNA genes, as well as the 5.8S. And these RNA genes we learned in the genome DNA lecture that come in tandem repeats. There are hundreds of copies of these repeats in the genome. And what you have here is, for example, the 18S, 5.8S, and 28S RNAs in the repeat. Now, at the upstream of this 18S is the promoter that specifically recruits RNA pol 1. And it has a upstream upstream element that binds to transcription initiation factors that are specific to RNA polymerase 1. And you can see that their names are kind of different, right? And they all have this one, TIF1A. And that one stands for working with RNA polymerase 1. Just like RNA polymerase 2, these initiation factors help put the RNA polymerase 1 in the right site here, and it starts transcription. So in eukaryotes, the RNA genes, the transcript contains all of these RNAs together. So there is a pre-RNA transcript that's made, that's very long, and then that long RNA gets processed by other RNAs into individual RNA molecules that gets folded and modified so that it has the certain structure that could be loaded onto the ribosome. Okay? So you first make a very long transcript. The RNA polymerase 1 makes a very long transcript, which then gets processed. It's the same idea for RNA polymerase 3. It makes the pre-tRNAs. These are longer RNA molecules than the actual tRNA that gets first transcribed and then processed and modified and turned into the tRNAs by RNA processing molecules. So in the case of the tRNA genes, we have the promoter, which contains the two sequences called box A and box B. And these two sequences are recognized and bound by a protein complex called TF3C. And again, that 3 stands for the initiation complex working with RNA polymerase 3. And analogous to what happens in POL2, you have these initiation factors interacting with the RNA polymerase 3 itself and positioning it at the right point so that it can start transcription and transcribe the tRNA gene. RNA polymerase 3 also transcribes the 5S RNA gene, so it's not the 18 and 23, but the 5S RNA gene. And in this case, it's also brought to the promoters, but in this case by a different sequence element. It's called box C. That again is recognized by the initiation factors that bring the RNA polymerase 3 there. RNA polymerase 3 transcribes other non-coding small RNAs. So for example, this is a U6 small nuclear RNA gene. And in that case, the promoter can also contain a TATA box and some other sequence elements. So in this case, these are different sequence elements that are recognized by a different set of initiation factors which interact with the RNA polymerase 3 so that it starts transcription at the correct start site. So again, what you see is that the promoters of certain genes contain DNA sequences that are bound and recognized by specific transcription initiation factors. And those initiation factors make interactions with a certain RNA polymerase, right? 1 or 2 or 3. And those interactions position the RNA polymerase at the promoter such that the catalytic site of the RNA polymerase aligns with the transcription start site, and that's where the transcription starts and continues. Any questions? All right, looks like I went a little bit fast, so if you have any questions, just come by here. And I'll see you in a couple of weeks.